Before we can start using Punnett squares to determine the outcomes of a mating event, we must first understand the dominant and recessive alleles. Now, what we see here are a pair of homologous chromosomes in a diploid individual and a number of genes that occur on these chromosomes. Now, genes can typically have two or more alleles that follow a dominant or recessive system. So what we see here is two alleles for each gene, one that is dominant and one that is recessive. And dominant alleles are indicated by the capital letter and recessive by the lowercase letter. So for instance, we see in flower color that we have a dominant allele coding for the purple color of flowers and a recessive allele coding for the white. Now, the idea behind a dominant recessive system is that if the dominant allele is present, that is the allele that will be expressed. So for instance, during transcription of gene expression, this allele will be the one that is chosen to transcribe, the purple flower color. Now, a recessive allele is only expressed if there is no dominant allele present, and therefore there is no choice. For instance, in seed shape, in this example, we have only recessive alleles to choose from, and therefore that will be the shape of the seeds will be wrinkled instead of round. So go ahead and pause the video in a moment and describe each of the outcomes, each trait for this plant, this hypothetical plant example that we have here. Describe the flower color, the seed shape, the seed color, and the pod color. Okay, so the flower color would be purple because there is a dominant allele present out of the two choices. The seed shape would be wrinkled because there is no dominant allele. Both alleles are recessive. Seed color would be yellow. Both alleles in this case are the dominant allele for the yellow color. And the pod color would be green because again there is a dominant allele present of the two choices. Now we need to understand the terms genotype and phenotype when expressing the alleles and the outward appearance of an individual. An individual's genotype describes exactly what alleles they have for a certain gene. So for example, in reference to the pod color gene, they could have two dominant alleles, two recessive alleles, or one of each. Now, when both alleles are identical, we call this a homozygous individual, homozygous for this trait. But we need to also further describe whether that means that they have both dominant alleles, in which case they would be homozygous dominant, or they have two recessive alleles, in which case they would be homozygous recessive. So both of these, both the symbol representation and the words used to describe the alleles, are examples of genotypes. The other genotype is the heterozygous genotype. In this genotype, the alleles are different, and therefore necessarily one is the dominant allele and the other is the recessive. Now, an individual's phenotype is actually the outward expression of that genotype. So what will the result be when you look at the individual? And of course, this follows that dominant recessive system we just described in the last example. So a homozygous dominant individual in this case would have the green pod color because the capital G is the dominant allele for green pods. The heterozygous individual would also have green pods because again it has at least one dominant allele in its genotype. However, the homozygous recessive individual would necessarily have yellow pods, the recessive phenotype. Okay, so we use a Punnett square to anticipate the probabilities associated with the genotype of the offspring created from a mating event. So let's do an example here using this Punnett square. Suppose we're going to cross two parent plants that are both heterozygous for pod color. So remember what this means at the molecular level. That means that both parents have in their original diploid cells two homologous chromosomes, one that has the dominant allele and one that has the recessive allele. That's what it means to be heterozygous for this trait. Now when they create gametes, and remember that both of these individuals have the green pod color because they have the dominant allele. When they create gametes, those homologous chromosomes are going to separate and turn into individual cells. Therefore, parent 1 can offer either the dominant or the recessive allele, and the same goes for parent 2. Therefore, we set these possible alleles donated from each parent up in the Punnett square, and then make the match between the possible combinations of these alleles. So when we do that, when we're combining, for instance, this allele from the one parent with the other, we get a homozygous dominant genotype in this possible outcome. When we combine a recessive and a dominant, this offspring is a heterozygous individual. And down here we see both of the recessive alleles have combined, making it a homozygous recessive individual. And then the phenotype of these individuals can be determined by the presence or absence of the dominant allele. So in this case, we would have 3 possible outcomes with the green pods and 1 possible outcome with the yellow pods. And we call this the phenotypic ratio of the offspring, a 3-to-1 ratio of dominant to recessive phenotypes. So each offspring here has a 75% chance of having green pods and a 25% chance of having yellow pods, each and every single offspring created by these 2 parents. So if 1,000 offspring were created, which plants typically do produce many seeds, we would expect 750 of them to have green pods and about 250 of them to have yellow pods. Okay, so now you take a turn. In pea plants, seed shape has 2 alleles. Round seeds are dominant and wrinkled seeds are recessive. So go ahead and cross a heterozygous plant with a plant that has wrinkled seeds. And remember to put the possible alleles from one parent across the top and the other possible alleles from the other parent down the side. And then answer these following questions. What is the probability that an offspring will have round seeds? And what's the probability of having a homozygous dominant offspring? So go ahead and pause the video and take a minute to work this out. Okay, so first you would put the alleles of one parent across the top. So we wanted to cross a heterozygous plant, and that means that they have 1 dominant allele and 1 recessive allele. So these are the possible gametes that they can offer. Now a plant that has wrinkled seeds is expressing the recessive phenotype, and the only way we can get the recessive phenotype is if we're homozygous recessive. So that plant can only offer recessive alleles, and you would put those 2 little r's down the side. So then you would go ahead and fill out those combinations in each of the squares of the Punnett square. And what we see then when we determine the phenotypes associated with each of these is that we have approximately half making the round seeds and half making the wrinkled seeds. So the chance of having round seeds is 50%, but homozygous dominant individuals are not possible from the combination of these 2 parents, and therefore the chance is 0% of having a homozygous dominant offspring.